I will share about meiosis. Meiosis is a process that produces gametes, such as sperm cells and egg cells. And what's the difference between meiosis and mitosis? As you can see, mitosis starts with the 2N DNA content and end with 2N DNA content, which means the only change is double of the replication of DNA content and back to the normal. And this is crucial for the growth and repair of the human or plant cells. However, meiosis drop down having two level of meiosis, and this is for the reproduction, and when the sperm cell and egg cell meet together, they back to the 2N DNA content, which can make a new baby. So this is the big difference between mitosis and meiosis. And I will talk about two stages of the cell division called meiosis 1 and meiosis 2. Meiosis 1 has 4 phases: prophase, metaphase, anaphase, and telophase. And meiosis 2 are the same. And it starts with a diploid cell, and from interphase, there's many chromatin. And the chromatin then will form together to form a visible chromosome. Visible chromosome then combined with corresponding homologous chromosome. And then in prophase 1, there's a important process called crossing over, which you can see the different color of the chromatid is not the same. And this create different gene combination and also create a large genetic variety. And why does it very important? Because, for example, if a person will die after drinking water, and with the same DNA content for the newborn babies and future generation will all die. And this is not ideal for the humans. And because of crossing over, it creates many different DNA from person to person, and this will solve this problem. This also explains why the same born siblings who born from same parents but having different looks. The paired up chromosome will line up at the equator. And for the anaphase 1, it will have the spindle fibers attached to the chromosomes, and the chromosomes will move towards the opposite pole. And for the telophase, the two cells will be formed, and keep in mind that sister chromatids are no longer identical because of the crossing over before. And moving to the meiosis 2, the meiosis 2 already turned into from the diploid cell into haploid cell, because there's only one set of chromosome. And the process are similar because they at the metaphase 2 they also line up at the equator. However, when the anaphase 2, it will not move the chromosome like the anaphase 1 before. Instead, it will attached to the chromatid and separated. And as you can see, there's a sister chromatid separated to move towards the opposite pole. And at the end of telophase 2, there's total 4 cells will be made.